see in microbial cultures. And there are three types of models to study that. First is thermostat, in which you are continuously adding and removing the new medium. So you have the volume constant, but the system is not constant. The other one is serial transfer, in which you take a fraction of the culture and then you inoculate this in a new medium, and you do that for every a time, during a very long time. So you take an liquid, you put in another medium, then you wait, you take another liquid, and you put in another medium. And finally, you have long-term cultures, in which you don't add anything after initial conditions and you don't remove anything. The only that you put is water to maintain the volume constant. But for that reason, we thought that long-term was a good study model to analyze diversity, because you are not losing any part of the system, so you can have more genetic diversity and also resource competition. So if we analyze, now we are focused on that type of culture, and if we analyze the dynamics of the population in that cultures, we can see that at the beginning, in hours, we have this exponential growth rate, and then a stationary phase that is typically where microbial people work in this phase, in both phases. But then, if we maintain the system without change, we can see a death phase in which the 90% of the population died. But then, the system maintains constant for a very long period of time. So the interesting thing is what happens here, how the population is maintained, even that you are not adding any resource of the system. So then experimental analysis show that first, at the beginning of this long-term stationary phase, we have periodic selection, in which mutants with a better fitness can invade the system, and so there is a competitive exclusion. But after 3 months, it is possible to observe coexistence of different morphotype of colonies. So the question here is, why emerge coexistence, and what is the mechanism involved in that? Yeah.
Can you explain the colors? What are the different colors like there?
Here?
Mm-hmm.
Okay. So in this case, after 3 months, they analyze the morphotype of colonies. So you can we should go to explaining in this lab. You can inoculate in agaroflake, and then you see that grow different morphotypes. So here, then you can, at different times, for example, after 1 week, after 2 weeks, you see the different morphotypes, and then you can do this plot. So it's the proportion of the different morphotypes in the animal.
So there's 4 different types.
Yeah.
Basically different phenotypes.
Yes, there are different phenotypes. So based on that, they assume that there are different in genotypes. Yes. Yes. For example, here, when you start here, you start with an isogenic strain. Yeah. Okay. So the question is, what are the mechanisms involved in this regimen shift between periodic selection and coexistence. And there are several mechanisms, but we focus on one, that is cross-feeding. And cross-feeding is the ability of one genotype to use metabolites secreted by other genotypes. So in this paper, the scientists discovered that first there are at least, they did this experiment in a chemostat, so it's a continuous medium, but they discovered that the presence of at least 33 intermediate of glycolysis and Krebs cycles. And also they saw that there are 3 mutants that coexist all the time. So when they analyzed that mutant, they discovered that they have difference in the mutants have differences in the growth rates and also in the rates to uptake and secrete different metabolites. So, for example, here you have this mutant that due to mutation now is possible to excrete glycerol, and this other mutant can consume that molecule. So this also can excrete acetate that can is able to consume for this other mutant. So this type of mechanism is called cross-feeding. I'm sorry?
Julian, does that at all include Julian atoms?
Yes. Yes. Yeah. Uh-huh.
They've actually got a large hemostat to hold Julian now.
Mm-hmm. So you know about the system? Yes. Okay. So we propose, this is a theoretical study. So we said, okay, we can have cross-feeding in this way. Here, these are genotypes, and these are different resources. So, for example, we can start with one genotype that consumes one resource, and then excrete another, and then it is consumed by another genotype, and so on. But in that type of regimen, we can observe periodic selection, because when this resource is depleted, then this genotype can coexist, can exist, so the other is advantage in relation. Do you understand? Okay. But then, if we observe some cycles, then we can have coexistence, because we are maintaining the system. Okay.
Can a genotype consume different resources?
I'm sorry?
Can a genotype consume multiple resources?
Yes. In the case of experimental analysis, they observed that the mutants can consume glucose, yeah, that is the medium that you add at the beginning. But also they can consume acetate or glycerol.
Yes, you assume that only
Here in the model, no. We are assuming only one resource per genotype.
Okay.
So I'm going to explain the model. But it's an agent-based model in which the probabilities that there are some probabilities that determine birth and death weight, and based on Monod equation to growth rate in bacteria. Also, we have a theory of resource allocation in which bacteria use more resource to reproduction when resources are in higher concentration, but then when resource is in lower concentration, they use their nutrients to maintain, to survive. And also, we consider a rate of resource consumption. So there is a relation between what do you need to grow in relation to maintain the system. And then, in terms of probabilities, we have three probabilities: one to survival, another to reproduction, and both depend on the resource, the relative abundance of the resource that genotype consumes. So every genotype can consume one resource and can excrete one resource. So we can see here the different probabilities that both depend on relative resource abundance. And also there are two constants that realize the value of resources needed to maintain the system in the 50%. Okay? So some assumptions of the model is the replication only depends on resource concentration, mutant rate is constant, the energy available in the system is constant, and there is not a space limitation. And the parameters, we have a limited pool of possible metabolites. So that value is n. And we start with a total abundance of resource at the beginning, and the constant that defines the probabilities, and the number of iterations. So, like a summary, we can see here the model. We start with the model with one type of genotype that can consume, in this case, A and J. No, I and J. I and J. Okay. With some probabilities, can maintain alive or dead, and it can consume this type of resource and can excrete this other type of resource, and then with this other probability can reproduce consuming different amount of resources. And then with PM probability can mutate. So one of the new bacterium has different genotype, and this different genotype is means that the new genotype can consume and excrete another type of resource. So it's clear until now?
Does it always change both things?
Yes. Yes.
Sometimes it changes one.
Yeah, no, in this case the model is like a double mutation because you are affecting both A and J. Yeah. I. So, there is the parameters. We use n equals 5. That means that there are 5 different metabolites possible in the system that are generated from the same genotypes. And we can see that at the beginning, it is possible to observe a periodic selection, and then there is a change to coexistence. And if we analyze the abundance by metabolite, we can see here that there is a very big fluctuation, but after is constant. Yeah.
Sorry, going back one slide.
Yes.
Each organism.
Oh,
sorry, 2 slides, I guess.
Yeah.
Consumes and excretes one metabolite. Is it possible for those to be the same?
No.
Okay, you block that possibility.
Yes, it's not possible.
Okay, that makes sense.
Yeah. So for that reason, we have n equals 5, and the possible metabolites is n times n less 1 times. Do you see?
Mm-hmm. Yep. Confident for it.
Okay. So here, the important thing is that the regimen shift between periodic selection and coexistence is related with equals the abundance of resources. So this is one thing that perhaps is necessary